This is the old university town of Göttingen that you see here. And it looked like that only a couple of weeks ago with the Christmas market. Very romantic, very German, very small city, a lot of good science. And it was a home base for me to go on this tour that we just heard about. And I could give my talk only about the tour. But let me just tell you, you see all the different cultures. I talked to people who spent years in Africa living with families in very remote areas, studying how the society evolves. I met people who tell me they are the first to get data from the new space telescope because they are the world experts in the search, in the search for extraterrestrial life. Crazy things. I've talked to people who told me that they spent time in southern Sudan. It's the youngest country on earth. Only 10 years ago it was established after long war times. And they are the world experts in international law, and they have educated the first judges in southern Sudan. This is all Max Planck. And yesterday I've seen the beautiful institute here, neuroscience. Again, different culture, different world. How does the brain function? I learned about the studies that are ongoing to understand how memory works. Why is it that it's suddenly more difficult to remember even a phone number, and it was easy before? What has changed in the molecules in our brain? Fascinating science. But one thing that I saw everywhere at all these different institutes, despite the different culture, is the shiny eyes of our young scientists. You talk to them and you ask them, what are you doing? What is the problem you're looking at? And they begin to tell you the story and how they got to it and why it's so exciting. So you see, they work with their hands, they work with their brains, but really they only work with their hearts in the end. It's passion. They really put everything into the science, and the best years that they have, maybe to do a PhD, to do a postdoc, the Max Planck Society is really almost like a jumping board, a springboard that takes you, up the next step on the ladder in your career. Today I want to tell you something very special that is very close to my heart, because I spent about 3 decades now working on a fundamental problem in biology, and that is how our genes actually work. Genes are silent, it's just information. So how can it be that with our genes we can generate an organism and we can keep cells alive, we can respond to the environment when we get an infection? It's all in our genes, but our genes, that's only information, it's silent information. The genes need to be given voice. And this happens in the process of transcription when mRNA is made. And it's made by a beautiful machine that is my favorite molecule and always will be. And it's called the polymerase. And I want to show you how that polymerase looks like and how it works, because it's copying our genes to produce messenger RNA. And you all know what messenger RNA is. Many of you have gotten it injected into the arm. Moderna, but also BioNTech, Pfizer, they have made vaccines during the pandemic that are based on messenger RNA. That's how powerful these messenger RNA molecules are. They can stimulate an immune response. They can really make our cells react. And this process of transcription, making these different messenger RNAs, is really at the heart of life. It underlies the development of organisms, the differentiation of cells, the formation of different cell types and different tissues. So what you see here is an embryo which is stained according to such gene activity in different colors. And you see that genes are switched on in different parts of the embryo, And why is that? It's because different types of cells need to be generated. Remember how life starts. You start with a fertilized oocyte. So you start with one cell where the genomes from the mother and from the father are combined. And from that one cell, within 9 months, you generate a human being through embryogenesis. And how does that work? It's because all the cells have all our genes. It's about 22,000 genes. But not all of those genes are active all the time. In the contrary, in a muscle cell, only those genes are active that you need to make a muscle cell. In a blood cell, those genes are active that you need to make a blood cell. So only a subset of our genes is active in these different cell types. And it's all controlled by transcription of our genes. And who's doing the transcription? It's the polymerase, and that's the central enzyme that controls which genes are active at which time and where they are active in our body. The genes are made of DNA, as you know, and then you transcribe them, you make a copy in the form of messenger RNA. And why is the messenger RNA so extremely powerful? It's because it's instructing protein synthesis. So the type of messenger RNA dictates what type of protein is made. And the proteins have this interesting property that they are all different. You know, DNA always looks the same. RNA is always a chain of nucleotides. But then the proteins are all different. And this is because the sequence of the gene is different. And then the sequence of the RNA copy will be different. And so you make a different type of protein. And these proteins, because they have different shapes, also have different functions. And this is why there's a protein that can make a muscle fiber. And another protein can make, that can transport, for example, oxygen in your blood cells. And another protein, for example, in your pancreas will be insulin. It's a hormone that can regulate your blood sugars. So those proteins have all those different functions, but they all come from DNA. But messenger RNA is the key. The messenger RNA that is produced will dictate which proteins are made. And, you know, it's nothing special about the proteins if you think about it. They're all made from the same amino acids. But because their shapes are different, they have different function. And we all know this because tools, for example, they're made from steel. They're all made from steel, but they have very different function. And why is that? Because they have different shapes. It's the same for proteins. Now, to catalyze this first step, to make this copy of our gene, to copy the DNA and make the messenger RNA, you need the polymerase enzyme. It's called RNA polymerase because it's making an RNA, a messenger RNA polymer. So you wouldn't believe how excited I was. You see how young I was at that time? You wouldn't believe how excited I was when I had my little baby in my hands, the polymerase enzyme. This was the first model, you see. And the company called me several times saying, it's impossible. We cannot make this model. It's impossible. It's so complicated. You know, all these chains intermingling. And then I told them, you also have to color them according to the code I give you. And so they made this first model of the polymerase. And I brought that home to Germany after 7 years abroad. And actually the last years we spent at California, where I've actually done this work together with Roger Kornberg at Stanford. Our son was born there. He's American. He keeps his passport. He wants to vote. He wants to be engaged. He feels American. Also, he has been back only a couple of times since. But that's how we are connected to the US, through that work, through family. And I want to tell you now how this enzyme works. Of course, I magnified it a little bit. In reality, it's only a couple of nanometers large. So what is a nanometer? It's very simple. You all know tennis balls, right? And so the polymerase enzyme, when you compare that to a tennis ball, right? The nanometer size enzyme and the tennis ball, It's the same ratio as if you would compare the tennis ball to the entire Earth. This is how small a nanometer is, just to give you an idea. And that's why, you know, we wanted to have a nice big model so that we have something in our hands to understand. How do you get a structure of that size? How can you solve such a complicated structure that is only a few nanometers? Well, you need big microscopes, like this one. This is where I did my PhD before I went to California. So this is basically where I learned to do structural biology. It's a synchrotron. It's about 1 kilometer around. It's in France, in Grenoble, the European synchrotron. And, you know, we took bikes to go around to do our experiments because such a big hall. And there you produce some of the strongest X-rays in the world. And such machines you also have in the US. We had one in Silicon Valley. Actually, there's two. There's one in Berkeley also. And I use those X-rays to shoot crystals of the polymerase, and that allows you to determine the structure if you work really hard and if you get lucky and if you make use of all the serendipity that you get. And then, you know, I took that back to Germany. I worked with my group for another 10 years to solve different snapshots. And, you know, like in the good old times of Hollywood, when you put the snapshots together to make a movie, we could actually make a movie of the polymerase, how it copies the gene and how it produces the messenger RNA molecule. So we look right into the center, into the active center of that machine. And in blue, you see the DNA. You see here the DNA duplex. And in red, this is the mRNA chain that is being produced. And you also see the enzyme moving forward. So this is a new building block in orange that will be added now to the growing RNA chain. You see now it turns red because it has been added to the growing messenger RNA chain. And then the enzyme moves, it moves to the next position because it needs to add the next building block. And can you believe it? It's doing this 3,000 times a minute. So the real movie, this has been slowed down dramatically. The real movie is going tuck, 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 tuck. You cannot watch it. That's why we slowed it down. So 3,000 building blocks a minute. This is happening as you sit here in all of our cells, especially in the liver. The liver is super active, right? It needs to make these enzymes to digest the food. Or if you drink a glass of wine afterwards, transcription will be very active. This will happening all the time because you need to make the enzymes to get rid of the toxic alcohol molecules. But there's a big problem here, right? We now know after these years of work how this copying machine works. But how does it actually find its target genes? As I told you, 22,000 genes. This is how the DNA really looks like. We have 2 meters of DNA in each and every of our cells. The cells are only a few micrometers, but each of them has 2 meters of the DNA thread. So how does the copying machine find the starting point where it should go and copy the gene? Over the last 10 years, we solved that problem in Munich and Göttingen to some extent. And I show you the solution. Actually, the polymerase is a very social molecule like we are tonight. It likes to work with other proteins. They help each other, and that's how they solve the problem. That's how they find the target gene. So there's all these little helping proteins here in red and yellow and green, and they will mark the position in the DNA where the polymerase should go. And you see now the big monster polymerase is coming, and it's finding its target site because there's these helper proteins, these colorful proteins that mark the start site of transcription. And now there's some other proteins and they kind of kiss each other above the DNA. This is because they retain the DNA. Once they got a grip on it, they don't let loose, you see. They found the spot and now they don't let loose. And there's another big protein, and this is needed because the DNA has 2 strands. You know, and if you want to access the information in the DNA, you have to separate the 2 strands. But how do you do that? You have to have a tool that introduces torque to open the DNA duplex, and this is happening with this big complex here. And then there's even larger one in blue up here. I don't even want to go into it because it's very complicated, but all of these proteins are assisting the polymerase, the copying machine, to find the start site of transcription, which is the beginning of the gene where you have to start to copy the gene. Now, how can you solve such structures, right? Because now we are talking again a different order of magnitude. And there, even these large synchrotrons wouldn't help you because you cannot even make crystals that you would shoot with X-rays. So thank God how it often works in science. When you're courageous and you start something, then somebody else is doing something that helps you. And in the meantime, over the last 10 years or so, there was a new type of detector developed for electrons. So now instead of using X-rays, we now use electrons. And they interact much better with matter. And we don't even need to go to the synchrotron. We can buy an electron microscope because the technology has advanced so dramatically. And actually today I saw one on campus on your neighboring institute. So that kind of work can be done here also on campus. This is, how these machines look like. It's about 5 million for one of those microscopes, 5 million euro, I should say, and a 4-meter-high machine. And so how do we do that work? Just very briefly, we take a droplet of protein solution, just a few microliters. We put it on this grid. This grid is about this, a few millimeters, 2-3 millimeters, and then we dump it. We don't freeze it, but we cool it down so rapidly that these protein molecules are now sitting inside of little holes on this grid held by surface tension. So that cryogenic sample can be imaged in the microscope. And you see here the little snapshots of the proteins. You can sort them in the computer, right? So now we make the crystal in the computer. We don't have to make the crystal in the lab. We make it in the computer. And then the resolution is very, very poor, but we just take millions of images, millions and millions. And then we take a supercomputer cluster and we put all these images together and we reconstruct an atomic structure. Now you may say, fine, they solved it, that's it. Let's listen to some music. But it's getting even more complicated. Everything I told you so far is actually totally simple compared to what the cell really does. Because when you look inside a cell, it looks like this. Nobody has seen it ever, so an artist drew it according to how we imagine it. It's an incredibly crowded place. It's almost like a liquid crystal inside the cell nucleus where the DNA is. So how does the polymerase go through, how does it run along the DNA? How does it go through the gene? I told you thousands of nucleotides a minute. Actually, human genes take 1 hour to be transcribed. 1 hour. That's how long the human genes are. Even at a speed of 3,000, 4,000 building blocks a minute, it takes 1 hour. And so how does the polymerase, in such an environment, it's almost like, it would snow, and then you would go through the snow at, incredible speeds, and just the snow would go everywhere. So it's a little bit like this. And so the polymerase, again, has a lot of helping hands, other molecules. And very recently, we published that only a couple of weeks ago. We made the first movie of how the polymerase goes through one of the building blocks in the cell nucleus. How can it, overcome these obstacles? There's one such obstacle here in front of the polymerase, and the polymerase is such a strong machine, it can generate a force. It can actually unravel the DNA from this disk, and it can displace proteins that are in the way. So it's a little bit like, you free the streets of new snow that has fallen so that the cars can pass. And often it also gets stuck, but then there's other proteins that can rescue it. It's like a fire brigade or so that comes, when you're stuck on the road. And especially what happens is that it goes forth and back. So these molecules, they fight against each other. The polymerase tries to go forward, but this kind of obstacle here, which is a so-called nucleosome, is pushing it backwards. But then there's this protein factor here from the fire brigade that can cleave off the RNA, and it can restart the polymerase. It helps it to try again. And eventually, the polymerase can overcome all these obstacles. Again, this movie is slowed by about a factor of 1,000. So you can imagine what's going on in the cell. Last thing I wanted to tell you is to make you aware that this kind of work can also be useful at some stage. Because so far, we learn how the process works, how messenger RNA is made, how our genes function, how they're activated. This is all great, we can teach it in school, we can teach it at university. But how do we benefit in the end? And what we find at the Max Planck Society, and many institutions have found that if you really generate new knowledge, I'm not talking about a few data points here or there, or do a measurement here or there. I'm talking about real knowledge, that our understanding is advanced. Then this new knowledge, at some stage, sooner or later, will be applied. It just is transformative knowledge. And so I give you one example, because we have been also working on the coronavirus. Because the coronavirus, of course, has the same issue, also has a polymerase enzyme. It's just a baby polymerase compared to my baby. It's more like a very small viral polymerase, but very powerful. It can amplify, not only amplify, it multiplies the RNA genome of the coronavirus. And we have been amongst the first 4 groups to solve the structure of the coronavirus polymerase. And actually we did that, in the spring of 2020. And the competitors were, of course, Chinese labs. So 3 Chinese labs did the exact same work, and all 4 laboratories published within a week or 2. That was a race, I tell you. In the night, I couldn't sleep much. In the night, I was writing a diary, because it was so exciting, this new virus, and we will probably be the first to see how this virus is multiplied, how it's replicated. And what was then exciting, that in the year after, we would be the first to also show how the first corona medication, remdesivir and molnupiravir, you probably have heard about it, because when Donald Trump got COVID, they gave him all that. It was not even approved, but he got the remdesivir as one of the first people. And so we actually showed later, after it was already used in the clinic, how it actually works. But that knowledge is now useful to find new antivirals, because this kind of medication is not working very well. It's has low efficiency, it has side effects, it needs to be applied by transfusion and so forth. So we have joined forces with chemists at another Max Planck Institute, and they screen 300,000 chemicals with a robot. And today, actually, I've seen such robots here. You have a wonderful facility also on campus to do this. And so we screen these hundreds of thousands of compounds, and we found actually a few new molecules that nobody has in hands yet that can stop the virus, at least, in the test tube. So we will explore that. Maybe one day we can make better medication based on this knowledge. I'm really grateful to the many young people with shiny eyes who helped me over the decades. And I'm really looking forward to my new job. It's a privilege to get to know people like David and many colleagues who do this wonderful job of not only doing great science, but also educating the next generation, that we have a great generation of scientists also in the future. You see, many people have passed through the lab, and they came from, I think, 35 different countries. In our lab right now, at one bench, we have 2 people, and one bench is actually shared by a Russian PhD student and a Ukrainian PhD student. And they go out together, and when they saw this thing happening, they looked at each other and are like, what is going on? This is has nothing to do with us. And this is also about science, that science, like sports, it brings people together. It builds bridges. And I think our world really needs to build bridges. We need that everywhere, within our countries, but also between countries. And that's what I wanted to tell you. Just that I'm at number 52 on my tour. Here, you see most of the institutes are within Germany, but we have 5 abroad. one in Jupiter. I think it's probably the location that cannot be rivaled, maybe by our Rome Institute, because when you go up the Spanish stairs, who has been there of you at the Spanish stairs? Yeah, many. You go up the Spanish stairs, you turn right, and it's just one corner and it says Max Planck Institute for the History of Art. And I really look forward to that one, believe me. Thank you very much for your attention. Thank you. Thank you. Thank you.